My name is Charlene Hurst, and I'm a graduate student at Oregon State University. I started my undergraduate college career as a music major and took one marine biology class, and that was it. I was hooked, and I've been doing science ever since.
Shasta is a parasite, something that fish don't like, causing disease every year. We have much to fear.
So the issue that I'm working on is if the 4 mid-basin dams are removed from the Klamath River, then Chinook salmon may be able to repopulate the upper basin.
2 articles demonstrate historic salmon runs to the Upper Klamath Basin before the Mid-Basin dams were built. J.B. Hamilton and colleagues, and Virginia Butler and colleagues.
And what we want to know are the effects of sea Shasta on salmon and trout, particularly since we now know that there are different genotypes of the parasite in the upper and lower basin. So in the Williamson River above Upper Klamath Lake, I sampled 20 sites using qPCR to assess the parasite density in the water, and I identified 2 areas of high parasite density. One was from the mouth of the Williamson River to the Sprague confluence, where we had a high proportion of genotype 2, and the other was above Spring Creek, where we also had a majority of genotype 2. We also have conducted sentinel fish studies, which resulted in no mortality in the Chinook and coho salmon, but resulted in 100% mortality in the non-native rainbow trout. And in the lab here at the Salmon Disease Lab, we've exposed juvenile Chinook salmon to either genotype 1 from the lower basin or genotype 2 from the upper basin. The lower basin Chinook exposed to genotype 2 survive from this infection, which means that they will likely survive being reintroduced into the upper basin. However, the lower basin Chinook exposed to genotype 1 in the laboratory died from this infection, which means that after reintroduction, the Chinook could bring the lower basin genotype 1 into the upper basin, and it could establish and therefore infect future runs of Chinook salmon.
Charlene is interested in the interaction of different strains within the salmonid host. For disease management, a possible use of this research would be to use less deadly strains like a vaccine to immunize the fish against the more deadly strains. While Charlene focuses on the Upper Klamath Basin, Matt Stinson is looking at parasite activity in other Northwest rivers.
So from the work that's been done in the Klamath, it's been very exciting, and so they wanted to know what else is going on in the Pacific Northwest, and so they've had me come out and sample throughout the rivers and tributaries in the Pacific Northwest. So I've been doing some juvenile fish exposures in some of these local rivers out here to see what the effects are outside of the Klamath River, but I've also been able to collect returning adult salmon, visiting hatcheries and taking samples from those fish to see what the types of the diseases those fish are getting. So my sentinel exposures have been taking place in the Willamette River and the Deschutes River of the Columbia River Basin, and for the returning adult salmon, I've been able to collect from all over the Pacific Northwest, like the Fraser River, the Cowlitz, the upper parts of the Snake and Columbia River, as well as down in Sacramento in the Sacramento River. So we want to know how this parasite has been affecting the other various salmonid species, for instance, the brown trout, brook trout, rainbow trout, cutthroat trout, steelhead, spring chinook, Atlantic salmon, and kokanee. So I've been able to do exposure with all these fish to see what kind of specific genotypes they're coming back with, and that's going to be some interesting research, we hope.
Earlier in this program, we mentioned 2 Klamath River characteristics that may affect polychaetes and parasite levels: mid-basin dams and the highly productive Upper Klamath Lake. Another characteristic of the Klamath River is its warm temperature.
My particular piece of the puzzle is focusing on climate change effects on disease caused by Ceratomyxa Shasta. Climate change is a really big idea. Kind of have to break it down into various processes that are a part of that change. Higher temperatures, lower summer flows due to reduced snowpack, earlier melting of that snow, more extreme precipitation events are all issues that could come about with climate change. On a micro scale, one of the things that could be affected by a change in climate, particularly higher temperatures and lower summertime flows, are areas in the Klamath River where the fish seek thermal refuge from the higher water temperatures. These areas provide a place for the fish to hang out when the main stem river temperatures are too hot. There's colder water coming in from tributaries. One thing that I'm trying to figure out is how these thermal refugia change over the summer and how severe the disease component is in these areas and how that will affect fish that are delaying their migration and prolonging their exposure to the parasite. On a wider scale, I'll be looking at the seasonal release of the parasite and what factors drive that release. So the Klamath River has a little bit more extreme conditions than other rivers in the Pacific Northwest, particularly in regard to its higher temperatures. As the climate changes, other rivers may trend toward the conditions of the Klamath. So if we can predict changes in the disease, we can be prepared and manage for disease effects in these other rivers where the parasite is currently present but not a problem.
You have met serious scientists pursuing a complex question. The Bartholomew Lab, a national leader in salmonid disease, is highly productive. While much has been accomplished, there remains much work to be done to bring Sea Shasta back into balance. We hope this program has brought you a better understanding of the scientific process and why careful science takes longer than the public sometimes wants. We also hope that the next generation will come to understand this kind of ecological complexity and take up the challenge for the future.